Our next speaker is Dr. Bose from the Brigham and Women's Hospital, who will be presenting fetal liver-directed gene editing results in supratherapeutic enzyme levels in a lysosome storage disease.
Hi, good morning. The focus of our lab's work is prenatal gene editing and cell therapy, and today I'll be presenting a part of our work focused on liver-directed gene editing in mucopolysaccharidosis type 1, or Hurler syndrome. We have no disclosures. This is a devastating disease which involves disruption of a lysosomal regulatory enzyme called IDUA. In the absence of IDUA, you get intracellular accumulation of glycosaminoglycans and multi-organ dysfunction. Kids are sick early in life, and most die by age 10. The standard of care is either enzyme replacement therapy or stem cell transplant, which are semi-curative but come with limitations in therapy, such as mortality, morbidity, and rejection. And gene therapy is a new focus which might mitigate some of these risks. And for diseases like Hurler's, in which pathology begins in fetal or early postnatal life, there may be an incremental benefit to prenatal therapy. Last year, our team demonstrated multi-organ system improvement in the Hurler mouse model by using a base editor, a molecule capable of performing a single base change. We observed high-level editing in the heart and the liver with resolution of cardiac pathology altogether, improvement in musculoskeletal pathology, and a strong survival advantage in treated animals. However, one limitation was that we saw very low-level brain editing, and we only observed mild neuroimprovement with no detection of significant IDUA in the brain. And this finding recapitulated a known limitation of existing delivery vectors to access the central nervous system without direct intracranial or intrathecal therapy. And so with this thought, and based on this finding, we explored a different approach with the thought that cranking up peripheral production of IDUA to supraphysiologic levels might compensate for limited diffusion across the blood-brain barrier. Our thought was to embed an optimized IDUA into the coding region of liver albumin, a highly expressed gene, to basically piggyback IDUA production on the native promoter. To do this, we used an editing technique using Cas9 to leverage intracellular repair machinery called homology-directed repair, or HDR, which allows for location-specific gene installation or integration. Importantly, HDR is limited in the postnatal situation due to its higher efficiency in replicating cells, and this is a main limitation for using HDR as a therapeutic. Therefore, our two hypotheses were that fetal HDR is more efficient than postnatal HDR, and that installing IDUA in the liver albumin locus might achieve our therapeutic goal. So to address our hypotheses, we performed two key experiments. First, we intravenously delivered non-secretory GFP to embryonic day 13.5 fetuses and day of life 0 neonates to compare pre- and postnatal HDR. We used either a liver-tropic AAV or a liver-tropic nanoparticle carrying the editing machinery, co-delivered with an AAV with our repair template, and this was to control for possible vector affinity differences to the fetus. Second, we delivered a secretory IDUA to the liver using HDR in postnatal mice to evaluate for enzyme activity. We used flow cytometry to quantify GFP as a proxy of successful gene integration and deep sequencing to measure overall genome cuts. We first wanted to account for any passive integration of our repair template in the absence of genome cutting, and so in the left panel you can see the so-called EFR control group, which had no GFP expression, suggesting that delivering a template of repair alone is inadequate for integration. We then saw that GFP expression and indels, or a word describing cuts, were highest in the AAV group, followed by the LNP group. And so with this validation in hand, we then compared integration neonates to fetuses to address our first hypothesis. These are some representative stereomicroscopic images taken 2 weeks after injection, and the gross appearance suggests that fetal integration was better than neonatal integration, agnostic of vector choice. On further analysis, we observed that the number of genome cuts was similar between fetuses and neonates, but that GFP expression was higher in fetuses by approximately twofold. Together, these findings implied that the integration of the homology template was more efficient in fetuses, thereby substantiating our first hypothesis. We then injected diseased neonates with the same editing machinery, but replaced GFP with secretory IDUA. And with 5% genome cuts, comparable to our initial study, we observed that supraphysiologic IDUA was achieved in the liver and other peripheral organ compartments, even compared to our prior work with base editing. And this supraphysiologic peripheral expression correlated with brain IDUA that was almost 7% of normal, and certainly greater than the 1% clinical threshold established for differentiating between severe and mild disease phenotype. Ultimately, based on the findings of our GFP study, we anticipate that fetal delivery may yield even higher levels than this, hopefully 2x. And so in conclusion, fetal homology-directed repair appears to be more efficient than postnatal repair, liver-directed editing leads to supraphysiologic enzyme in disease-relevant peripheral compartments. And finally, supraphysiologic systemic enzyme increases central nervous system IDU activity to clinically relevant levels. But at the end of the day, who cares? And I would submit that this peripheral production approach is something that may facilitate rapid scale-up to many diseases that require enzyme or a missing protein, particularly those without a dominant mutation that's targetable by way of a specific genomic technique. And as we believe that prenatal therapy is not only viable, but potentially the best option for diseases with an early phenotype, we're engaged in further work to see if these effects are then magnified with fetal delivery. There's a large team behind me, and I appreciate all their support, Dr. Paranto himself, and all of you for your time. Open to questions.
Thank you. We'll start with some questions.
Hi, King Win, Cohen Children's. Thank you very much. I had done some work with this with Tippy McKenzie, UCSF, and I appreciate that you've increased the systemic levels of your enzyme and that was able to penetrate into the brain. Since you're addressing this problem at a time when the barrier is still open, would it be another angle to directly address the brain cells themselves? We found that microglia are the responsible cells for many of these lysosomal storage disorders. Could you directly then use gene therapy to just directly edit the microglia themselves so that you have a lasting production? Since the enzyme that does make it in gestation might, expire in a few weeks, and you won't be able to get more enzyme in.
Yeah, it's hard to hear. I think you're asking, is it possible to edit microglia themselves to then have them migrate to the brain and produce enzyme? It's a valid hypothesis, and a lot of people are working on that. There are a couple limitations in that. One is the question of, are cells that you're transplanting fetally actually going to differentiate into microglia? Are they real? And I know Dr. McKenzie's done some work on that. The second is the efficiency of that technique, which is still in question. And the third is that there's a couple of trials out there right now, phase 1, phase 2 human trials, looking at lentiviral modification of ex vivo microglia. But there's potential benefits to doing gene editing itself in that you don't have a foreign cell then coming in and migrating to the brain. And you also achieve systemic therapy, which is an important part for a multi-organ disease.
Other questions? I have one, really elegant work. I'm curious about the sustainability or the durability of the increased enzyme activation. Is it something that's fleeting, or do you feel like it lasts for a while?
The sustainability of
the enzyme activation.
Yeah. So our prior work showed that if you can get enzyme production in utero, it persists all the way through 6 months. And that's really the appeal of this over other enzyme therapeutic approaches. You're actually getting the cell to produce the enzyme. You don't have degradation of the insert, and it's there forever as long as those cells are replicating and survive. There is theoretically the chance that you hit a population of cells that don't persist into adult life, that are preferentially fetally oriented and therefore die off. But in our prior work, we showed that we're actually able to have preferential targeting of stem cells in the liver that basically allow you to have proliferation for a lifetime, and particularly in settings. So some people have demonstrated that if you do a partial hepatectomy with administration of this kind of therapy in the liver, those stem cells can then proliferate and regenerate the liver.
Fantastic. Great work. Thank you.
Thank you.